Come to biology, the seventh study of life. Morphogenesis in animals involves specific changes in cell shape, position, and adhesion. Now that we have learned about the main events of embryonic development in animals, we will address the cellular and molecular mechanisms by which they occur. Although biologists are far from fully understanding these mechanisms, several key principles have emerged as fundamental to the development of all animals. Morphogenesis is a major aspect of development in both animals and plants, but only in animals does it involve the movement of cells. Movement of parts of a cell can bring about changes in cell shape or enable a cell to migrate from one place to another within the embryo. Changes in both cell shape and cell position are involved in cleavage, gastrulation, and organogenesis. The cytoskeleton, cell motility, and convergent extension. Changes in the shape of a cell usually involve reorganization of the cytoskeleton. Notice this figure. Consider, for example, how the cells of the neural plate form the neural tube. First, microtubules oriented parallel to the dorsal ventral axis of the embryo apparently help lengthen the cells in that direction. At the dorsal end of each cell is a parallel array of actin filaments oriented crosswise. These contract, giving the cells a wedge shape that forces the ectoderm layer to bend inward. Similar changes in cell shape occur during other invaginations, inbuctings, which means, and evagenations, outbuctings of tissue layers through development. The cytoskeleton also drives cell migration, the active movement of cells from one place to another in developing animals. Cells crawl within the embryo by using cytoskeleton fibers to extend and retract cellular protrusions. This type of motility is akin to the amoeboid movement. But in contrast to the thick pseudopodia of some amoeboid cells, the cellular protrusions of migrating embryonic cells are usually flat sheets, lamellopodia, or spikes, phlopodia. During gastrulation in some organisms, invagination is initiated by the wedging of cells on the surface of the blastula. But the movement of cells deeper into the embryo involves the extension of phlopodia by cells at the leading edge of the migrating tissue. The cells that first move through the blastopore and up along the inside of a blastocell wall drag other behind them, thus helping direct movement of the entire sheet of the cells from the embryo's surface into appropriate locations in the blastocell. The involuted sheet then forms the endoderm and mesoderm of the embryo. Notice this figure. There are also many situations in which cells migrate individually, as when the cells of a somite or the neural crest disperse to various parts of the embryo. Cell crawling is also involved in a convergent extension type of morphogenetic movement in which the cells of a tissue layer rearrange themselves so that the sheet becomes narrower, convergent. Notice that. While it becomes longer, extends, as shown in this figure. When many cells wedge between one another, the tissue can extend dramatically. Convergent extension is important in early embryonic development. It occurs, for example, as the archenteron elongates in sea urchin embryos and during involution in the frog gastrula. In the latter case, convergent extension is responsible for the change in shape from the spherical gastrula to the submarine shaped frog embryo. Notice this figure. Roles of the extracellular matrix and cell adhesion molecules. Scientists are coming closer to understanding the signaling pathways that trigger and guide cell movement. The process appears to involve the extracellular matrix ECM, the mixture of secreted glycoproteins lying outside the plasma membranes of cells. is known to help guide cells in many types of morphogenetic movements. ECM fibers may function as tracks, directing migrating cells along particular routes. Several kinds of extracellular proteins, including fibronectin, promote cell migrating by providing specific molecular encourage for moving cells. The experiment depicted in this slide, as shown in this figure, provides evidence for this function of fibronectin in guiding cell migration. Evidence also exists how fibronectin plays a similar role at the leading edge of the involuting tissue during gastrulation. Fibronectin fibers line the roof of the blastocell, and as the future mesoderm moves into the interior of the embryo, the cells at the free edge of the involuting sheet migrate along the fibers. Researchers can prevent the attachment of cells to fibronectin by injecting embryos with antibodies against fibronectin. This treatment prevents the inward movement of the mesoderm. Other substances in the ECM keep cells to the correct pathways by inhibiting migration in certain directions. Those depending on the substances, they secrete non-migratory cells situated along migration pathways may promote or inhibit movement of other cells. As migrating cells move along specific pathways through the embryo, respiratory proteins on their surfaces pick up directional cues from the immediate environment. Such signals from the ECM environment can direct the orientation of cytoskeleton elements in a way that propels the cell in the proper direction. Also contributing to cell migration and stable tissue structures are glycoproteins called cell adhesion molecules, CAMS, which are located on the surfaces of cells and bind to CAMS to other cells. CAMS vary in amounts, chemical identity, or both from one type of cell to another, and these differences help regulate morphogenetic movements and tissue building. One important class of cell-to-cell adhesion molecule is the cadherins, which require calcium ions for proper function. There are many different cadherins, and the gene for each cadherin is expressed in specific locations at specific times during embryonic development. Researchers have vividly demonstrated the importance of one particular cadherin in the formation of frog blastula. Cadherins are also involved in the tight addition of cells in the mammalian embryo that first occurs at the 8th cell stage. When cadherin protection begins, as we have seen, cell behavior and molecular mechanisms contribute to the morphogenesis of the embryo. The same basic cellular and genetic processes ensure that the various types of cells end up in the right places in each embryo. The developmental fate of cells depends on their history and on addictive signals, coupled with the morphogenetic changes that give an animal and its spores their characteristic shapes. Development also required the timely differentiation of many kinds of cells in specific locations. Two general principles integrate our current knowledge of the genetic and cellular mechanisms that underlie differentiation during embryonic development. First, during early cleavage divisions, embryonic cells must somehow become different from one another. In many animal species, initial differences between cells result from the uneven distribution of cytoplasmic determinants in the unfertilized egg. By partitioning the heterogeneous cytoplasm of a polarized egg, cleavage parcels out different mRNAs, proteins, and other molecules to plastomers in a type of asymmetrical cell division. The resulting differences in the cell's cytoplasmic composition help specify the body axis and influence the expression of genes that affect the developmental fate of the cells. In amniotes, local environmental differences play the major role in establishing early differences between embryonic cells. For example, cells of the inner cell mass are located internally in the early human embryo, whereas trophoblast cells are located on the outside surface of the blastocyst. The different environments of these two groups of cells appear to determine their very different fates. Second, once initial cell asymmetries are set up, subsequent interactions among the embryonic cells influence their fate, usually by causing changes in gene expression. This mechanism, termed induction, eventually brings about the differentiation of the many specialized cell types making up an animal. Induction may be mediated by diffusible chemical signals, or if the cells are in contact, by cell surface interactions. Fate mapping. Researchers have worked out the development history of every cell in Canorhabditis elegans, beginning with the first cleavage division of the zygote, as shown in this figure. Although such a complex cell lineage has not yet been determined for many other animals, biologists have been making more general territorial diagrams of embryonic development called fate maps for many years. In classic studies performed in the 1920, German embryologist W. Vogt charted fate maps for different regions of early amphibians embryos. Vogt's results were among the earliest in layers created by gastrulation is traceable to cells in the blastula before gastrulation has begun. Later, researchers developed more sophisticated techniques that allowed them to mark an individual blastomer during cleavage and then follow the marker as it was distributed to all the mitotic descendants of that cell. Developmental notice fate map of a frog embryo. Developmental biologists have combined fate mapping studies with experimental manipulation of parts of embryos, in which they study whether a cell's fate can be changed by moving it elsewhere in the embryo. Two important conclusions have emerged. First, in most animals, specific tissues of the older embryo can be attributed to certain early founder